If we look back at the Miller-Urey experiment, one of the things that we saw was that it produces amino acids, and we just went through all the stuff about how you can take amino acids, polymerize them, and make them into polypeptides. If they get long enough to fold up, then they become proteins. But that's not the only thing that they got. They got other types of things. And one of the things that they got were molecules of this sort. Now, this is a very highly stylized version of this molecule, but it's got three parts to it, and that's what this picture is trying to show. This part here, which is the central core, is a sugar that's made out of 5 carbons. And the way this is drawn, each one of these lines here represents a covalent bond, but we're drawing it now in a way that is done typically in organic chemistry to make it easier to sort of understand. See the vertex right here in this pentagon right there, that corner. That is a carbon. That represents a carbon. This represents a carbon, this represents a carbon, that represents a carbon, that represents a carbon. So every time we draw a line and it has a turn to it, we see in our mind a carbon there. And all of them, again, are covalently bonded. You notice that they form kind of a ring, this pentagonal ring with the oxygen right here. Now that's an oxygen, obviously not a carbon. And if you remember, oxygen can form 2 covalent bonds. There's 1, there's 2, so that's fine. Carbon, remember, forms 4. So the question is, where are the 4 carbon covalent bonds here? Well, this carbon right here is going to have 1 that goes up to this nitrogen, 2, 3, and it seems to be missing 1. It's not really, because again, in organic chemistry, we don't draw all the hydrogens. We just assume that all the extra valences, all the extra covalent bonds that can be formed are filled by hydrogen. So we see in our mind a 4th bond right there that forms a hydrogen. Let's look at this carbon. This carbon goes down here, and it's got a hydrogen here, and this one's not showing it as an oxygen and hydrogen here. I'm going to show you the detail of this in a little bit more strict detail here in a moment, but right now we just see this as a sugar. Now attached to this sugar, coming off of this side of the oxygen, is a nitrogenous base, and this is a base that, again, it's nitrogenous because it has nitrogen in it. And on the other side, there's this. Now this is a carbon here, and it's the 5th carbon that's in the sugar. And coming off of that carbon is this group right here. And if you look at this carefully, you might recognize that this is phosphoric acid, except it's not. It's missing the hydrogens already. So this is the conjugate base of phosphoric acid, and it's called phosphate, PO4. Okay, so this sugar then has 2 things attached to it. This 5-carbon sugar has a nitrogenous base attached to it and a phosphate group. Whenever you see that pattern, sugar, nitrogenous base, phosphate group, 5-carbon sugar, then we're looking at what's called a nucleotide. And in particular, to be specific, it's called a nucleotide monophosphate. But I'll get into that in just a little bit more detail later in the course. All right, so this is the kind of pattern that we are getting out of a Miller-Urey type experiment. And so who cares? Well, this one is important. You can even see already there's an important sort of root here, nucleo, referring to the nucleus of a cell. The reason it's called that is because we find this in the nucleus of the cell, and in fact we can do the same thing with this that we did with the protein, with the amino acids. We can polymerize them and make a polynucleotide, except we don't call it polynucleotide. I'll talk about that here in a moment. Let's look a little bit more at this sugar though. I want to talk first of all, what is a sugar? What's a carbohydrate? One of the things I want you to be aware of is this. Scientists use the word carbohydrate slightly differently than nutritionists do. To a nutritionist, a carbohydrate is what a scientist would call a polysaccharide, a complex carbohydrate. To a scientist, a sugar is any carbohydrate, including simple sugars and disaccharides, monosaccharides, polysaccharides, all those things are what a scientist would refer to as a carbohydrate. So we are going to take a look at this one carbohydrate because it gives us a sense of what carbohydrates really are. Now this is the same molecule that I showed you before. And it has the oxygen here. And remember I said each time that there is a little vertex, each time there's kind of a corner, there is a carbon. So here are the carbons written out. Also remember I said that coming down here, 1, 2, 3, 4 valences. I said we don't see that hydrogen. We see it in our head, but we didn't draw it. Here it is drawn. This is the thing that's going off the nitrogenous base. If this were just the sugar, this would be an oxygen-hydrogen group, just like here and here. The oxygen-hydrogen, remember, comes down, the carbon bonds to the oxygen, and then the oxygen, its other valence bonds to the hydrogen. We don't draw the line there. But this sugar right here, notice how many carbons it has. 1, 2, 3, 4, 5. And notice how they're drawn. Here's the ring. The oxygen is the top part of the ring. And you notice this carbon and this carbon are both attached to the oxygen. But notice that it's asymmetrical. This one does not attach to any other carbon except the one in the ring. But this one attaches to a carbon in the ring and a carbon outside the ring. The carbon outside the ring is the carbon that we define to be the number 5 carbon on this sugar. Okay, and so we then number the carbons backwards, 5, 4, 3, 2, 1. Now in the lab, I've asked you to draw a sugar. When I asked you to draw the sugar, I also said, please number the carbons correctly. This is what I mean. We need to get that carbon that's outside the ring is the last carbon. So on this particular type of sugar, it's carbon number 5. On glucose, it would be carbon number 6. Now the point is that we just number them backwards. Now for those of you who have had some organic chemistry, here's how the numbering scheme actually works. A straight chain form, this is not the only form that you have of this molecule, but the straight chain form has on one end what's called an aldehyde group. The aldehyde is always into carbon number 1, and then we just number from the chain following down from the aldehyde group. Okay, but notice then, it has 5 carbons, and then how many oxygens? Well, here it's showing that it's attached to something. If this were just the sugar, this would be an OH group, and if this were just the sugar again, this would be an OH group. So we would have O here for the OH, 2, 3, 4, and then a 5th oxygen here. So notice the number of oxygens is the same as the number of carbons if you do it this way. How many hydrogens? Well, there would be an OH here, so there'd be 2 hydrogens, one on that OH and one here, 3, 4, 5, 6, 7, 8, 9, and then an OH 10. Notice that's twice the number of carbons. Okay, so that is typical of carbohydrates. The carbohydrates typically have the same number of carbons as they have oxygens, and then they have twice as many hydrogens. And that's why we can write carbohydrates in this formula right here, CH2O. And then this N represents that pattern is repeated some number of times. In this case, N is 5, because it's 5 carbons, 5 of those, 5 oxygens, and 10, 2 times 5, hydrogens. Glucose, C6H12O6, same sort of thing. We could be CH2O to the 6th. So that's why this is called a carbohydrate. You see it? That's carbo, that's hydrate. I'm not going to look it up on Wikipedia, but that's the carbo, and that, notice, looks like water. It is. So it's carbohydrate, water. Whenever you see that kind of pattern, carbon has approximately the same number of oxygens and approximately twice as number, now I say approximately, it doesn't have to be exact like it is here, but approximately twice as many hydrogens, then we're looking at a molecule that's a carbohydrate. That's the structural formula. That's our definition of it. Okay, so that forms this central core of the nucleotide. Now we have a nitrogenous base that comes off and the phosphate group that comes off. The various nitrogenous bases can be these for the nucleotides that we are interested in. These aren't the only bases that you can have on here. For example, we'll see another one that's nicotinamide. But the point is that each one of these then forms this kind of a ring. Sometimes it's a double ring. And they have on them a nitrogen. They have always a nitrogen, and it makes it a base. So that's why we call it nitrogenous bases. This one is called cytosine. This is uracil and thymine. All of these form what are called pyrimidines, and then the purines are guanine and adenine. Again, remember, these are not the only types of bases that can go here on carbon number 1, but they're the important ones for what we're going to be talking about here in just a moment. All right, so those are various bases. The phosphate, again, is PO4, which is the conjugate base of phosphoric acid. So this is an acid. It's also a base. Notice we saw that pattern before. Amino acid, same thing. One side is an acid, the carboxyl group is an acid, the amine group is the base group. So this is an acid, but it also has a base associated with it. Okay, so we can polymerize this. I promised you we could do that. So we can. What we can do is this. Here is our nucleotide. There's the base, and there's the OH, and there's blah, blah, blah. And this is carbon 1, 2, 3, 4, 5. We can take another one, and if we orient it in the same direction, so that the phosphate group, carbon number 5 phosphate group is sticking up, then we can take the oxygen and the hydrogen off the phosphate. Now notice this phosphate is drawn as it's ionized here, and here as if it's not ionized. But we imagine that that hydrogen is there, because it is in paper chemistry. We take the hydrogen off of here, and we attach then this phosphorus through this oxygen to this sugar. So it goes like this. Carbon number 5 on the bottom nucleotide attaches to the phosphate group. The phosphate group then attaches to carbon number 3 on the next nucleotide up, and we've made what's called a dinucleotide. So this bond is a phosphate bond. It's a phosphate group that connects these two different molecules. And notice it's the same sort of thing. Here's a monomer, here's a monomer. We've now made a dimer, 2 pieces together. This particular bond that connects 2 nucleotides together is called a phosphodiester bond, or a phosphodiester linkage. Those of you who've had organic chemistry, all of you essentially probably will have organic chemistry, will learn that this is an ester group because it has something bonded to a double bond oxygen, and then it's connected. So there's an ester group over on this side, there's an ester group on this side, and this has phosphorus instead of carbon, and so we call it a phosphodiester. Now, if you didn't follow all that, don't worry about it. Memorize the name, phosphodiester bond. That's connecting two nucleotides together. Okay? Who cares? Well, you should care, because these phosphodiester bonds, when we repeat them over and over again, we make a polynucleotide. But polynucleotides have a special name. They're not called polynucleotides. They're called nucleic acids. You've probably heard of that before. Well, let's take a look. If we connect these together and continue to connect them, I can make a big long chain. And these chains can be huge. They can be millions, literally, of nucleotides long. And this particular one right here is 1, 2, 3, 4 nucleotides. And the bases, notice the bases all stick up here, up to carbon number 1, and every single one is connected carbon 5 to carbon 3 through these phosphodiester bonds. If I make this polynucleotide out of this type of sugar right here, then I have what's called ribonucleic acid, RNA. That's how I make RNA. So we got this out of the Miller-Urey experiment. We got these simple building blocks for RNA. Now remember, RNA is important. We'll see why later on. But this right here then makes RNA inside your body. Now in your body, the particular types of bases that you can find on RNA are going to be these: adenosine, cytosine, guanine, and uracil. The thymine is not there. So here, this one, cytosine, uracil, guanine, and adenine are what we tend to find on RNA. And the interesting thing about this is that when RNA forms, it doesn't actually look like this picture. This picture is a little bit misleading. It shows that the bases are all sort of in the same plane, and that's not right. That is not what they actually look like. Here's what I want you to imagine. Here, my fist is the sugar, and my thumb is the phosphate, and my finger is the base. The base. So now what I'm going to do is I'm going to attach the next one up. Okay, so again, I have here the sugar. Okay, is my fist. This is the phosphate, and there's the base. Now in this picture, it's showing them all basically in the same plane, but that's not what they are. They're not like this in the same plane. In reality, they're twisted like that. There's an angle, and so that the bases come off at an angle. And then the next one goes, and it comes off at another angle. Next one goes, it comes off at another angle. And so what you should see in your mind is not this nice flat linear structure, but a spiral going up. The bases form a spiral, like a spiral staircase. And that's called a helix. And it forms this one helix, a single helix going up. All right, so that's ribonucleic acid. But sometimes you can make nucleotides out of a different type of sugar. And when you do that, then you get something slightly different. Now take a look at this. Look at these two things and tell me, what is the obvious difference that you can see here? Well, again, here's carbon number 2. Notice it's carbon number 2 now. On ribose has an OH group. And on carbon number 2 on this particular one does not have an OH group. It only has H. So this is identical to this. The left-hand one is identical to the right-hand one except for one thing. The left-hand one is missing an oxygen here in carbon number 2. So therefore, we call it missing oxygen ribose, deoxyribose. Now, we can still make nucleotides out of deoxyribose, but if you do that, you get a slightly different kind of spiral, which is extremely important in making the overall structure of the molecule, which I'll talk about here in just a moment. All right, so the key difference then of not having that oxygen on carbon number 2 in deoxyribose is that spiral that I was talking about that it makes, like this and this and this, and goes and spirals around, is at a different angle, set of different angles. And what that allows then is that you can have a nucleotide, polynucleotide of deoxyribose like this. Now, if you look at this as a slightly different arrangement that we saw before, you see the ribbon, the red ribbon here going down this one side actually represents the sugars. So if we go back to here, you'll see this is sort of what's represented by the red ribbons, and then the things sticking out are the bases here. So we go here, these are the bases, there's T, G, and so forth. Now, what happens then is I can make a polynucleotide with deoxyribose, and when I do that, one of the key differences is that it doesn't have in its group of nucleotides uracil. It has thymine on its bases instead of uracil. It still has cytosine, guanine, and adenine, but this is only found in DNA, and this is only found in RNA. That's why we see the T, for example, here. But here, then you have the phosphodiester bond here, phosphodiester bond here, phosphodiester bond here, and so forth, and we have this sequence of bases. And it is forming a spiral. It's just like we saw with the RNA, except the spiral has a different angle. But then I can take another one, and something really interesting happens here. We'll study this in great detail later in the course, but I can take another polynucleotide, and if it has the right sequence of bases, such that wherever there's a T on the left-hand nucleotide, there's an A on the other, and then wherever there's a G on the left hand, there's a C on the other, then the two nucleotides that are swirling around can actually swirl around each other and form this structure, which is called a double helix. Now, a double helix is almost never represented correctly in structures that you find on the internet. If you Google DNA structure, the vast majority of pictures you're going to get are wrong, because what they show is a ladder that's twisted, and that is not what DNA is. It's not a ladder that's twisted. It's two spiral staircases wound around each other. And this picture here is far better than what you tend to find on the internet. It shows that the structure has additional structure on top of just this twisted ladder. You see, right here, this is a really small groove, but on the other side is a big one. And then here, there's the small one again, and then a big one. And if you see this in 3 dimensions, then this small groove actually swings back behind here, comes around here, and then it swings in front and then goes back behind. And this big one goes the other way. This is coming in front, goes back behind, then comes in front. This big groove we call the major groove, and this is called the minor groove. Twisted ladders don't have that. Twisted ladders just simply have one groove running around each side. But the big one here, the major groove and the minor groove, are important for us, because if we're using our detailed understanding of how DNA operates, we have to keep in mind where the minor and major grooves are. Because, for example, proteins, enzymes will bind to DNA. How they bind to DNA depends on where the major and minor grooves are. So this is something that we have to know. I also want you to start to get a sense of the size of this molecule. Now, notice each one of these things is a nucleotide. And how many nucleotides can I have? Well, I can have a lot of nucleotides. I can have millions of nucleotides on one of these molecules. So the molecule can be very, very long. In fact, literally a single molecule of this could be a few centimeters long. Take all of the DNA in your cells, one cell, take all of its DNA, the entire DNA is going to span about 1 meter, and I'm not exaggerating. It's 1 meter long. So these molecules are extremely long, but they're also extraordinarily thin. If we look here, from here to here, the width of this spiral staircase wrapped around another spiral staircase is about 2 nanometers. Now make sure before you move on, you know exactly what I mean by nanometers. 2 nanometer means 2 billionths of a meter. 2 times 10 to the minus 9. In other words, take a meter stick and chop it up into 1 billion pieces. How long would that take you? Well, if you do the calculation, if you chop a piece off every second and never stop, every single day, every single week, every single year, for as long as it takes to chop it up into 1 billion pieces, it's going to take you over 32 years to do that. That's how many pieces it takes. That's how many 1 billion actually is. Take 2 of those, and that's the width. It's really, really tiny. Now, one complete swing of this, one complete twist of the DNA is exactly 10 of these bases. So 10 bases go up, and that gives you a complete swing of one complete spiral. Okay, and then the distance then between each of these bases from here to here is, as you can see here, 0.34 nanometers, which means an entire twist is 3.4 nanometers long. Right? So there's more to it as well. Notice this is going in one direction, 5' to 3'. This is going in another direction, 3' to 5'. That has to do with the way the molecule is oriented, and we'll get into that detail later on in the course when we talk specifically about the structure of this. What's important for us now is to understand this. The DNA and the RNA both have sequences of nucleotides, and therefore sequences of bases. So when we see this, we talk about the sequence of DNA, we talk about T, G, G, T, T, G, C, blah, blah, blah. Now, we always read this, and again, I'll get into this more detail later on, but we always read these sequences from the 5' end to the 3' end. So when we talk about the DNA sequence on this strand here, we're talking T, G, G, T, T, C, G, C, not T, G, T, G, T, C. Okay? Now, this strand would be the other way. Since, again, we always read 5' to 3', it would be reading ACACA, not ACCAAC. Okay? So that's the structure. The structure of DNA, again, is not represented very well on most sources in the internet that I've seen. I want you to have the real structure in your mind, and that is not a twisted ladder. It's two spiral staircases wound around each other with a minor groove and a major groove. Okay, so that's DNA. This is RNA. Those are the main differences between the two. Now remember, when we looked at the total amount of dry mass in your body, the majority of your nucleic acid was in fact RNA. DNA only forms a small part of your total mass when you don't have any water. And the reason is the RNA has actually got to be doing a lot of stuff. The RNA is far more active in what it does than the DNA is. The DNA is quite passive. Really what DNA is a way of storing information. But RNA, and especially proteins, are the things that take that information and do stuff with it. That's why there has to be more of it. So when we look at these things bound together, remember I said that if you have the right sequence, you'll be able to get these things to bind together. And this is what we get. Here then again is the phosphodiester bonds between two adjacent nucleotides. And here's a guanine, here's an adenine. Now on the complementary strand, that's the strand on the other side. We call this is the strand and this is its complement. When we look at the complementary strands, if we have this sequence where every place where we see a G, we have a C versus an A and a T, those things will be able to wind around each other. Otherwise they don't. They don't wind very well. So these base pairing rules are essentially that. G binds with C and A binds with T. But also notice, how are they bound? Well, look here. This is a polar covalent bond, and this is a polar covalent bond, and this is a polar covalent bond, and these are also polar. And wherever you have a negative charge here, you have a positive charge here. So these charges are opposite and attracting. So these sort of dotted lines are representing hydrogen bonds. These hydrogen bonds then are what hold those two strands together. Now, notice that when you have guanine and cytosine bound together, you get 3 hydrogen bonds. When you get adenine and thymine, same sort of thing, except you only get 2, right? That's real. That's not just some artist's idea. So this guanine-cytosine bonds are slightly stronger than the adenine-thymine bonds, and it matters. If you look at small prokaryotes that are growing in the really, really hot water in Yellowstone National Park, all these really hot geothermal springs, you'll see that they are enriched in CG pairs compared to us. And that's to hold their DNA together to be able to handle the heat. Okay, so this is then the key difference between DNA and RNA. DNA has deoxyribose instead of ribose. It forms bases with ACG and T, not ACG and U. And when it does form, it tends to form a double helix. Now, it isn't true that RNA will not form a double helix. It will, but it's not as stable. And when it does form a double helix, it tends to form a double helix with itself. So it looks like this. This is what RNA tends to do. You'll get a double helix it'll form with a major groove and a minor groove and so on. But it'll tend to do it with itself, and it won't do it for very many bases in a row. And that's because the angles don't quite fit. The physics doesn't quite work for a really, really long, stable molecule. RNA and DNA can also form a double helix together, in which case then you get a hybrid nucleic acid. We'll see examples of those later in the course. But these things will form, RNAs will form double helices, but typically only with themselves or with DNA. Okay, so the last step in all of this is what's the connection between all of these things? We've talked about the Miller-Urey experiment giving you the basic building blocks of proteins and nucleic acids and so on. How are these all structured together? This is one of the major themes of this course. They're structured together in the following way. The DNA, as you know, is generally found in the nucleus, not all of it, but some of it, most of it's found in the nucleus. And it holds information. That information is read and copied into RNA. The RNA then leaves the nucleus, goes into the cytosol, and gets read by some structure, which we'll discover here shortly is called a ribosome. And that information in the RNA is used to build a protein. And this structure right here is amino acid, and you see the protein folding into its tertiary structure. This idea has been called the central dogma. It's inappropriately named. The guy who named it, Francis Crick, he claims anyway, didn't quite understand what the word dogma meant. But at any rate, this is the central dogma of biology, is the information in DNA is copied into RNA. That information then is used to build protein. So information flow in cells goes from DNA to RNA to protein. They're all functionally connected. And it folds back on itself, like we saw in this pie chart here. What is it that the proteins are doing? Well, the proteins are partially doing signal transduction, but look at this, nucleotide metabolism. The proteins are controlling nucleotide metabolism. So there's the feedback. Nucleotide goes to another nucleotide, goes to protein. The protein controls what the nucleotides are doing. So all of this is a giant cycle. RNA gets its information from DNA, protein gets its information from RNA. The DNA operates to make the RNA based on the proteins. We're going to put all of that together. In fact, that's going to take up the majority of our efforts in this course to understand exactly what's going on with all of that.